Good morning, children. This is Venkatachalam, your science 2 teacher. Children, today we are going to discuss the practical portion in our science, in particularly BIO-BOTANY portion, exercise number 8, 9, 10, 11. Alone we are going to discuss. First one, photosynthesis. Test tube funnel experiment. So what are the required materials? What is the aim, procedure and observations? We see one by one. What is the aim of this experiment? To prove the oxygen is evolved during the photosynthesis. What is the aim? To demonstrate that this oxygen is evolved during photosynthesis by a test tube funnel experiments. What are the requirements? Number one is glass tube, test tube funnel, beaker, pond water, hydrola plant. The procedure: the glass beaker is filled with 2/3 of pond water, in which a small amount of sodium bicarbonate is dissolved. A glass funnel is placed in a beaker that encloses the tube of a hydrola. A glass test tube is filled with water, is kept inverted over the stem of the funnel. Now the apparatus is kept in a sunlight for 4 to 6 hours. Children, the next one is observation. We can observe a steady stream of bubbles rising up from the ends of hydrola branches is kept within the funnel. This gas is collected within the test tube by the downward displacement of water. This gas is tested for the oxygen. Can you understand? The gas bubbles are liberated from the hydrola plant. It reaches a test tube and it displaces the water downwards. They take the test tube and keep the burning stick near the mouth of test tube. The increased flame will appear. So it proves that during the photosynthesis, oxygen is evolved. Okay, children. Question number 2. Parts of a flower. The question is to dissect and display the given flower in calyx, corolla, androecium and gynoecium. Draw a neat label sketches. So what are the materials are required? Flower, needle, blade and paper. The procedure: with the help of the needle, dissect the given flower, Hibiscus rosa sinensis, and the floral parts are calyx, corolla, androecium, gynoecium. The calyx and corolla is a non-essential whorl or auxiliary whorl. The androecium is a male part of the flower. The gynoecium is a female part of the flower. Both are involved directly in the reproduction. So it is called a essential whorl or essential reproductive organ. Children, draw and label the parts of flower and dissect and display in order: calyx, corolla, androecium and gynoecium. Okay, take one network, tuck in LSS and arrange in order: calyx, corolla, androecium, gynoecium. Okay, children. Question number 3. Mendel's monohybrid cross. In genetic portion, the aim is study the monohybrid cross by using a model or picture or photograph. Find the phenotypic and genotypic ratio of given plant. The Mendel proposed 3 different types of law: law of dominance, law of segregation, law of independent assortment. The first law of dominance: when 2 homozygous individuals with one or more set of contrasting characters are crossed, the characters that appear in a F1 hybrid are dominant, and those who are not appear in F1 generation, it is called recessive. It is explained by a checkerboard method. So the definition is: a cross involving one pair of contrasting characters is called monohybrid cross. The procedure: Mendel take a 2 homozygous tall plant and a pure tall plant cross with a pure breeding of dwarf plant, and producing gametes. The gametes are fused together and producing a F1 generation. All the F1 hybrid plants are tall. Again, F1 generations are self-classed and they result in the tall and dwarf plants in a F2 generation, and it given a result phenotypic ratio is 3:1, 3 tall and 1 is dwarf. The genotypic ratio is pure tall 1, hybrid tall 2, pure dwarf is 1. Children, question number 3. Anatomy. The stem and root of the plants are made up of a different type of tissue. This tissue form a different layer, the composition of stem and root. So study of the internal structure of a stem and root, in particularly monocot root and dicot root, monocot stem and dicot stem. So it is essential to get familiarized with a sectioning and staining technique used with the plant material. It is also necessary to take the section with uniform thickness so that the light passes through the stem equally and get a different tissue from the material, and the clearly visible one under the microscope to examine the tissue clearly. It is desirable to stay in a section with a suitable stain as a different stain color. Children, question number 11. Observation of transverse section of dicot stem and dicot root. Aim to observe the TS of dicot stem and TS of dicot root from a permanent slide. Identification. The given slide is identified as a transverse section of dicot stem. Reason. The ground tissue is differentiated into epidermis, cortex, endodermis, pericycle and pith. The epidermis single layer compactly arranged one, multicellular epidermal hairs are present. Cortex, it is a broad one, heterogeneous cortex, and vascular bundles are arranged in a ring one. It is a character of dicot, conjoined, collateral, endar and open vascular bundle, and 3 to 6 layer of parenchyma tissues are present in a hypodermis. The protoxylem is towards the pith. It is called endar protoxylem. Broad pith is present. These are a character of dicot stem. Next one, dicot root. The given slide is identified as a TS of dicot root. The reason, the vascular bundle or radial vascular bundle and a single layer of epidermis, unicellular root hair is present, homogeneous cortex, and xylem is exarch and tetrarch. The Casparian strips and passage cells are present in endodermis. Cortex is made up of a parenchyma cells. These are important reasons to confirm that the given slide is dicot root. Okay children, let's continue the remaining portion in next class. Take care. Bye.